Hello and welcome to this session of ACS Science Talks, Connecting the World Through Science. This is the virtual lecture series, Scientific Talks by specialists on specialized topics for a specialized audience. I'm Kunal Gupta and I'm pleased to be your host for today's broadcast of ACS Science Talks. I'm joined by my colleague, Dr. Mihir Jha and Dr. Manoj Gogoi, who will be moderating today's live Q&A. Let me introduce our speaker for today. Our speaker today is Professor Yoichi Hoshimoto. Professor Hoshimoto received his MSc and PhD from Osaka University under the supervision of Professor S. Ogoshi in 2013. He then joined the Frontier Research Base for Global Young Researchers, Osaka University, as a tenure-track professor from 2013 to 2018. Then he was promoted to Associate Professor in the Department of Applied Chemistry, Faculty of Engineering at Osaka University in 2018. Since 2023, he has been recognized as an outstanding young researcher in the Center for Future Innovation, Faculty of Engineering, Osaka University. To date, he was selected as the 2019 Early Career Advisory Board of ACS Catalysis and a member of Scientific Advisory Board of ChemArchive. His recent research interests include homogeneous catalysis with transition metal and main group element complexes, as well as development of original N-heterocyclic carbenes. Thank you, Professor Hoshimoto, for joining us. The stage is all yours.
Thanks. So let me share my slide. Okay. Does it work well?
Yes, it's working.
Okay. Thank you for very kind introduction. I'm very happy to have this opportunity today for sharing our chemistry with many participants. Before talking my chemistry, let me introduce myself a little bit more. Okay, so I was born at Nagano Prefecture, locating at the almost center of main island of Japan. Nagano is truly countryside, and I was very active for winter sports. So we had Winter Olympics in 1998. It was really famous. And then I moved to Osaka, which held World Expo 1917, and will organize Expo 2025 again. So if you have any interest, please visit Osaka at that moment. Anyway, I studied my PhD thesis under the supervision of Sensei Ogoshi. Then I started my independent academic career at Osaka University from 2013. And I had also wonderful experiences at Queen's University, Canada, under the supervision of Professor Cassie Craden, who is now head of ACS Catalysis. And now I'm joining to Professor Lucas Gussen's group in Uo University at Bofen. So I'm going to talk from Germany today while fighting with slight jet lag. But I am really happy if you can enjoy our team's chemistry. Okay, I'm gonna talk about mainly two topics. One topic is the Lewis Basic project, Lewis Base chemistry, using our original hot spin oxide introduced N-heterocyclic carbene, namely toxins. Then I will move to the second topic, which is a Lewis acid project using trial rings. So let me start the first topic. Of course, you know, the concept of Lewis acid and bases play a key role when considering the chemical bonds. Generally, reaction between Lewis acid and bases give isolable and self-stable classical Lewis adapt, CLAs. However, the reactivity of original component is quenched. On the other hand, when Lewis acid and bases include sterically bulky substituents around their reaction site, the formation of CLAs are inhibited by steric repulsion. In such cases, weakly coordinated association complex called frustrated Lewis pair, FLP, is generated. FLPs can show a high reactivity and hence has been applied for many molecular transformations. However, isolation and storage of FLP species are always very difficult. Unattractive reactivity of FLP is, needless to say, heterolytic cleavage of H2, because such H2 activation was predominantly based on the use of transition metal sulfur. For example, Stefan and co-workers nicely demonstrated that reaction between this T-butylhophine and tris-pentafluorphenylborane BCF smoothly gives this FLP species in situ. And this FLP rapidly mediate the heterolysis of strong HH bond. As mentioned, we cannot isolate and store this FLP because this tends to easily decompose by nucleophilic addition of this phosphine to the para carbon in BCF. In other cases, reported by Stefan, mixing 2,6-lutedyne and BCF gave equilibrium mixture of these species. And this FLP can activate H2, but again, FLP species cannot be isolated, and in addition, difficult to control interconversion between these species. Given these backgrounds, we envision the generation of both CLAs and FLPs by using a single pair of Lewis acid and base. If such system can be achieved, we can generate reactive FLPs from isolable, self-stable CLAs somehow. So how do you realize it? Our idea is very simple. Introduction of substituent having both small and large unit in Lewis base. When small substituent direct toward Lewis acid, as you can see, you can make such very stable CLAs here. On the other hand, when bulky comes to the front side, then steric repulsion generates to give FLPs. And if we can induce their interconversion using external stimuli, then we can generate FLPs whenever we want. Then what molecule we should design to realize this picture? Our answer is this N-heterocyclic carbene. N-hospine oxide substituted imidazolidine called Toxin. Synthesis of toxins are very easy. Treatment of this imidazole with hospine chloride generate this species in situ, and without isolation, in situ oxidation gave this stable, air-stable toxin HOTF salt. Then deprotonation using potassium butoxide afforded toxin as a crystalline solid storable under inert gas atmosphere. These are solid-state structure of toxin HOTF and toxin determined by XRD analysis. In toxin HOTF, oxygen and hospine oxide oriented to the syn direction with respect to this NP bond. On the other hand, in this toxin case, these two atoms adapt anti-orientation. So in this case, more open, three-dimensionally open, wide reaction field exists around the carbene-carbon, while in this case, very limited, narrow field is available due to the large T-butyl groups. Thus, reaction field around this carbene-carbon should be expanded or shrink largely through the rotation of N-hospine oxide. In fact, we experimentally and theoretically confirmed that rotation of this hospine oxide can easily proceed at room temperature with the activation barrier about 10 to 14 kilocalmol. But toxin predominantly exists as this anti-conformer to avoid lone pair repulsion between carbene and oxygen. Okay, next we explore the reaction between toxin and BCF. And this carbene boron CLA was isolated at room temperature. Monitoring the reaction at -90 degrees clarified the generation of oxygen borane CLA, and then converted into carbene borane species through the rotation of hospine oxide. When using para-protonated analog of BCF, we could isolate this oxygen boron CLA and fully characterize by XRD analysis. These results showed Toxin includes two Lewis basic site, hard phosphine oxide and relatively soft carbene. And the coordinating phosphine oxide occurs first to give this kinetic CLA and then transformed into thermodynamically stable carbene-boron adduct. This is the molecular structure of carbene-boron adduct. And as you can see, phosphine oxide rotates nearly 170 degrees from anti-free state to syn conformation. We quantitatively evaluated that reaction field around the carbene was drastically expanded over 20%, and thus Toxin can accept such huge Lewis acid to make stable adduct. This guy is air and moisture stable white solid shown here. And you can store this for years, over years, without significant decomposition, even in the rainy season of Japan. So we can expect this adduct is almost quenched. Indeed, no reaction proceeded when H2 was pressurized into the DCM solution of this adduct. If heterolysis of H2 takes place, then we can say active FLP species can be generated at room temperature. But it didn't happen at room temperature. On the other hand, once heated the system at 60 degrees, H2 activation proceeded quantitatively, probably by a generation of this FLP species through the rotation of phosphine oxide. To get insight into the role of phosphine oxide, we prepared this corresponding phosphine substituted carbene-boron adduct and confirmed no reaction occurred at 60 degrees. We also carried out this reaction at 100 degrees, but only decomposition of this molecule was observed. Thus, we can say this oxygen should play a key role in the generation stop of FLP from CLA. Due to the time limitation, I don't mention details about mechanism, but based on mechanistic studies, including stoichiometric reactions, kinetics, and DFT, we currently propose this reaction path. From this thermodynamically stable CLA, first, thermally induced transfer of borons from carbene to oxygen takes place. So boron jumping happening. Then rotation of phosphine oxide separate boron and carbene, and dissociation of this OB bond and recombination gives FLP. Okay, so let me summarize this topic. We have developed N-phosphine oxide substituted imidazolidine toxins and demonstrated thermal responsive interconversion between stable carbene-boron CLA and reactive FLP species using characteristic rotation of N-phosphine oxide unit. Now let's turn our focus to the other interesting reactivity of toxins. As you can see, toxins include both nucleophilic carbene and electrophilic phosphorus within a single molecule, but it's stable enough at the ambient conditions. So we envision the application of this ambiphilic reactivity for molecular transformations. What we did is one-part transformation of gaseous CO2 into unsymmetrical ketones. This is a picture of this first step reaction between toxin and CO2. NHCs are well known to react with CO2, and similarly, toxin smoothly scavenges the CO2 to form this imidazolium carboxylate intermediate. Then, addition of this carboxylate oxygen to phosphorus center and cleavage of this NP bond concertedly proceeded to give this anhydride, mixed anhydride. Of course, this guy can be isolable, but without isolation, in situ methylation gives this key intermediate. including two leaving groups. So it means imidazolium and phosphate on this CO2-derived carbon center. When you can add, then you can add two carbon nucleophiles step by step without any cryogenic conditions, producing unsymmetrical ketones. Of course, you know, CO2 can be easily converted into many organic molecules, such as esters, carboxylic acids, carbonates, carbamates, by using heteroatom-based nucleophiles. However, one-pot introduction of two carbon nucleophiles is very challenging. So yeah, we believe Toxin did a nice work to demonstrate the first example of conversion of gaseous CO2 into unsymmetrical ketones. Okay, so next, let me briefly introduce the application of toxins as a mouse-dentate ligand for transmetals. As mentioned, toxin includes these two conformers, anti and syn. So metallation can generate a variety of coordination complexes. In fact, several groups, including as demonstrated, toxin worked as an attractive platform for preparing a variety of metal complexes. And we also focused on the dynamic behavior of phosphine oxide unit. For examples, we demonstrated pressure-swing, reversible CO chemisorption, carbon monoxide chemisorption on zero-valent nickel center based on the interconversion between these two toxin nickel carbonyl complexes. Generally, dissociation of CO from low-valent metal center rarely proceeds at such a mild conditions due to the strong metal to CO back donation. But in our case, hemilabile coordination ability of N-phosphine oxide assisted the substitution mechanism for CO coordination and the dissociation processes, which eventually established room temperature reversible CO chemisorption on zero-valent metal center. Moreover, we use this ionic liquid having nearly negligible vapor pressure as a dispersant of toxin nickel complexes, and thus this system can be reused several times. What we saw interesting in this system is CO adsorption, in this way, CO adsorption takes place in solvated state, homogeneous solution. But CO dissociation would predominantly proceed on solid state. This is fine crystals are generated here. So solution state reaction and solid state reaction. So we discussed such details in this paper. Okay, then we envision what's going to happen if we add second metals. Because toxin includes two characteristic Lewis basic site, we envision toxin can also work as a good platform to prepare hetero bimetallic complexes, which can undergo dynamic conformational change through the rotation of phosphine oxide. For instance, we recently demonstrated a strategy for reversibly modulating the coordination environment around the metal center through the Lewis acid-mediated interconversion between syn bidentate toxin dicarbonyl or 1,4-diene complex and anti-nickel aluminum heterobimetallic complex. This result presents a complementary approach to the well-accepted Lewis-based mediated substitution strategy, but it's totally different. Lewis acid-mediated repelling out one coordination site. Okay. We also confirmed toxins afford heterobimetallic copper aluminum complexes by selective one-pot dual metallation processes and the subsequent rotation of phosphine oxide trigger the transmetallation between these two component species. So in this case, the rotation of phosphine oxide in toxin ligands can be used for precisely programming the approach and separation of two metal centers by preventing their random collision, often induces the decomposition of hetero-multimetallic complexes. Okay, let me conclude the first topic using our original NSC toxins. We also developed phosphine imide substituted derivatives called PIMM. Then you can introduce such chiral and amino acid derivatives on the NSC molecules. If you have any interest, please check our papers. Okay, so let's go on to the next topic, Lewis acidic trial catalyst project. Yeah, here is very dry. Very dry, yeah, sorry. Okay, this is the simplified scheme for the current production of H2 from carbon-based materials. At this moment, we are using fossil fuels and natural gases for the production of H2, but I believe in near future, biomass and food waste, livestock waste, and wasted plastics, all those hydrocarbons can be used for H2 production as well, based on the waste to hydrogen strategy. In these cases, before production of H2, crude hydrogen, including the contaminants such as CO, CO2, CH4, H2O, blah, 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 is generated. So we must remove all these contaminants before the use of H2 in the hydrogenation of unsaturated molecules, such as nitrogen for ammonia production, CO2 for methanol production, or fuel cells. However, in such hydrogenation reactions, we currently use transition metal catalyst. And these transition metals can be easily deactivated by those contaminants. So the H2 purification process is very important to prevent the deactivation of catalysts. However, such processes consume a lot of energies, time, and human resources, and it loses H2 itself. That's why many scientists are devoting their effort to optimize and modify the efficiency of these processes. Against these situations, we envision the direct use of crude hydrogen for the hydrogenation of unsaturated molecules. If this achieved, we could completely bypass these problematic purification processes. To this end, we need CO and CO2 tolerant catalyst. And we thought it should be achieved, it should be possible by using main group elements. So in this context, we are now focusing on the hydrogen storage using liquid organic hydrogen carriers, LHCs. Hydrogenation of unsaturated molecules using purified hydrogen gives saturated products. These products can be applied for the storage and transportation of H2. And dehydrogenation regenerate the unsaturated molecules and H2 again. For example of homogeneous catalysis, Yamaguchi and Fujita reported the hydrogenation of 2-methylquinoline in the presence of purified hydrogen and subsequent dehydrogenation from tetrahydroquinoline using this iridium catalyst. BCF was also applied for the hydrogenation of 2-methylquinoline under diluted conditions using purified hydrogen. In addition, BCF catalyzed dehydrogenation was also reported. However, the optimal reaction conditions, especially the concentration of substrates, are totally different between these two subsystems. Thus, so far, subsequent hydrogenation and dehydrogenation catalyzed by main group element has been limited to our system shown later. So our target is crude hydrogen. If we can use crude hydrogen for hydrogenation step, we can simultaneously separate and store H2 in HOLC molecules. Then subsequent dehydrogenation will produce highly purified hydrogen. In other words, previous system can only work for H2 storage, yet our target system should be also applied for H2 purification. So we started our experiment. In the presence of 1 mole% catalyst, hydrogenation of 2-methylquinoline was carried out in toluene. When using supercatalyst, which is well-known hydrogenation ruthenium-based catalyst, reaction of course proceeded quantitatively with purified hydrogen. However, when using 1,2,1,2,1 mixture of crude hydrogen, H2CO2, reaction did not proceed well. We also explored rhodium-Wilkinson catalyst, homogeneous iridium catalyst, ruthenium macho catalyst, and heterogeneous palladium carbon catalyst. But in all cases, hydrogenation did not proceed under the crude hydrogen conditions. So we turned our attention to test BCF, which generates this type of FRP in situ. But again, the hydrogenation was significantly suppressed under the crude hydrogen conditions. Yeah, this can be expected because FRP have been reported to react with syngas, COH2, and almost all reported FRP can react with CO2. So to achieve hydrogenation under the crude hydrogen conditions, we must optimize the boron catalyst to prevent an irreversible quenching of FRP species by CO and CO2. Okay, so here showing selected result for boron optimization and start from this BCF. Okay, substitution of para-fluorine atoms in BCF drastically improved the reaction efficiency. So modulating electrophilicity of boron atom is very important. And interestingly, substitution of 1-pentafluorobenzene ring with pentachlorobenzene ring slightly improved the reaction efficiency. This strategy is modulating front strain. In per-molecular repulsion between Lewis bases, such as quinoline, tetrahydroquinoline, CO, CO2, and H2O, including these gases, we never dehydrated gases, so H2O is included. So combining electrophilicity strategy and front strain strategy, this boron developed by Source and co-workers afforded the tetrahydroquinoline in 75% yield under the crude hydrogen conditions. But we could not improve result anymore based on front strain strategy. Thus, we needed to develop a novel strategy. So we focused on the modulation of remote backstrain, intramolecular repulsion within triaroboron. Okay, so structure of free triaroboron is always planar. But after the coordination by Lewis bases, the geometry changes into the tetrahedral. Okay, then intramolecular repulsion, so steric and electronic repulsion between remote substituent generates, and go back to the plane structure with promoting the dissociation of Lewis bases. What we did is the substitution of meta-fluorine atoms with chlorine, bromine, and bulky aryl groups. Especially in the case of chlorine and bromine, catalyst activity was improved. So, in order to precisely evaluate the catalytic activity, we carried out a reaction under neat conditions with crude hydrogen in the presence of 0.1 mole% catalyst. It's crazy, right? Under such extreme conditions, BCF shows only 11 TON, so turnover numbers, catalytic turnover number, only 11. But meta-fluorine boron exhibited 1,000 TON, and chlorine 4,000, and bromine over 5,000 TON. Interestingly, Lumo energy level of these boranes are nearly identical. Thus, these results should be caused by the steric factors of meta-substituent. Moreover, in the presence of 0.005% bromine substituted Sb third and purified hydrogen, TON reaches to the nearly 3,000, showing high potential of main group elements as a hydrogenation catalyst. Okay, finally, we demonstrated a proof of concept for H2 purification. First, we carried out hydrogenation under crude hydrogen conditions in an autoclave reactor. Then open its autoclave reactor valve and pumping up all CO and CO2. Then close the valve again or transfer the solution to the another vessels, heating 200 degrees. We found this borane also catalyzed the dehydrogenation from here under the neat conditions. Thus, H2 and 2-methylquinoline was obtained again. So through this single reaction cycle of hydrogenation and dehydrogenation, H2 purity successfully and drastically increased from 30.3 to 99.999%, over 99.999% purity. Okay, so let me summarize topic 2. We applied our original trial boranes for hydrogenation of quinolines and 2,6-lutidines by directly using crude hydrogens. We believe these results nicely show the power of main group catalyst beyond its simple alternative for transformer catalyst. Okay, today I shared some of our recent achievement on Toxin project and trial borane project. Due to time limitation, I have omitted mechanistic details and some important references. You can find such information from our papers shown here. And these works has been accomplished by my team's ingenious collaborators, members. So I'd like to say my deepest thanks for their tremendous effort. Okay, please give me time to advertise International Kyoto Conference on New Aspects of Organic Chemistry, ICOC 15, which will be held in this November at Kyoto. On the behalf of organizing committee, I am happy to welcome you to join ICOC 15. Okay. So let's scan now, everybody. Yeah, this invitation is a part of important task of mine today. Okay, scan now. If you have any interest, please feel free to ask me details. Okay, so finally, thank you very much for your kind attention, and I'm happy to answer your questions. Okay, that's all.